the Actica-1 trial, which compared capecitabine. So this was kind of amended. Initially, it was observation, but when capecitabine became standard of care, it was then capecitabine versus gemcitabine and cisplatin. I think this trial has finished recruitment quite a while ago now. I would have always thought that we would have seen some results by now, but thus far, we unfortunately have not. There's one additional trial that I would like to mention in the adjuvant setting, which is the ARTEMIDE trial. And ARTEMIDE is a platform that uses rilvegostomig. Rilvegostomig is a bispecific targeting both PD-1 and TIGIT. If you're a more pessimistic person, you would probably say, well, the success of TIGIT was kind of limited in trials that we've seen across different cancer entities thus far. Now let's hope that this co-targeting bispecific strategy will pan out in the end, because you will see throughout the next couple of minutes that there is a lot of trials that actually try to count on rilvegostomig. So ARTEMIDE biliary 01 is the adjuvant trial that is using rilvegostomig as an immunotherapy in combination with chemotherapy, and the chemotherapy can actually be investigator's choice. It's a placebo-controlled phase 3 trial aiming to recruit, or having recruited actually 750 patients, at least in Germany. I know this trial is closed and I think it's fully recruited, but it will probably take some time until we actually see the results. So the assumption is Actica will probably report soon, but ARTEMIDE, it's going to take some time, but it seems to be fully recruited by now. In the scenario of targeted therapy in the adjuvant setting, I'm going to skip this slide because John Bridgewater is going to tell you more about it in just a little while. Now, if you're staying in the earlier or the more localized CCA scenario, of course, radioembolization is an interesting topic. And in our last webinar last September, we talked about the IMUY phase 2 trial, which is a trial aiming at patients with unresectable intrahepatic cholangiocarcinoma. It is a phase 2 trial that has two experimental non-comparative arms combining radioembolization with either durvalumab or durvalumab and tremelimumab, basically similar to the STRIDE regimen in HCC. And I'm happy to say that by now this trial has been fully recruited with 50 patients. And what I would also like to point out that this trial has been going on for quite a while. It's really struggled regarding recruitment. It was only recruiting in Germany initially, but then to enhance recruitment, Spain basically joined in, and this really sped up the recruitment phase. I think it's one of those good examples showing that we really need to work together across not only different centers, but also across different countries. So hopefully some results along the way for the IMUY trial. Staying in the field of radioembolization, there's also a very, very interesting trial ongoing in the neoadjuvant setting, actually. And Julia Adeline is the PI here. And Julia, you have to excuse me. I'm sure I'm mispronouncing your trial. I'd call it Sirocho, but you may want to correct if you're on the call. So Sirocho is actually using radioembolization in this neoadjuvant setting, targeting resectable intrahepatic cholangiocarcinoma. So the IMUY trial was unresected. This is now really aiming at resectable and in the neoadjuvant intention, and it's combining actually the standard of care for the adjuvant, the capecitabine, which is split up 4 cycles capecitabine around the third, and then 4 another cycles of capecitabine in the adjuvant setting. And the comparator arm is what we currently usually do, surgery, and then adjuvant capecitabine for 8 cycles. This is a trial that, to my knowledge, is currently only recruiting in France. I'm sure that most of you have seen these data and have read the publication. I think that was quite exciting for our community. The NeoGOLP trial that was just recently, I think last week or 2 weeks ago, published in the New England Journal of Medicine. And I think there are not that many manuscripts that are actually published for biliary tract cancer in New England Journal. It's might be a game changer, and I think it's really leading forward in the ballpark of perioperative therapies and neoadjuvant therapies. The trial included 9-week course of neoadjuvant therapy with a bit of a combination that we are not that used to, gemcitabine, oxaliplatin plus lenvatinib plus a PD-1 antibody, then resection, and then capecitabine, and the primary the primary endpoint was the event-free survival, which you can see has been reached. This is an intermediate analysis, and the overall survival has also been shown. Thus far, I think formally it did not reach statistical significance yet, but there will more analysis will be coming. And I would like to mention that also in a neoadjuvant design, there is the NeoLangio trial, which is a phase 2 trial that has just now started to recruit in Canada. But it will also open in Spain, to my knowledge. This is kind of a joint venture between Arne Vogel and Gonzalo Sapizoshin. And when Gonzalo moved from Canada to Spain, he basically took this trial with him. So I think this can only be applauded to do it again across 2 different countries. The design of the trial is that you have the neoadjuvant treatment actually again with a quadruple combination, including immunotherapy, in this case gemcis, Abraxane, and rilvegostomig, then surgery, and then an adjuvant course. And the immunotherapy is going to be continued for up to 12 months. So this is a trial, again, in the very promising setting of neoadjuvant and perioperative therapy that has just now started recruitment. We're now moving to the more advanced setting of biliary tract cancer. And again, there is rilvegostomig that we will encounter also in the advanced stages quite frequently in the next couple of months and years, I guess. There are some data for rilvegostomig in the palliative setting in combination with gemcitabine and cisplatin. This is from the Gemini platform that is testing basically different types of bispecific, rilvegostomig being one of them. And 30 patients in this phase 2 trial, single-arm phase 2 trial, received this combination of chemotherapy and immunotherapy. The results are as follows. I always have a bit of a hard time interpreting too much into those single-arm phase 2 curves. If you want to compare it to our standard of care regimens, I think it is quite similar in terms of median PFS and overall response rate. But again, these are only 30 patients. And we will see how this combination will perform in a phase 3 setting. And this is exactly what the Artemide biliary 02 is testing. So just to not confuse, biliary 01 is the adjuvant setting, and Artemide biliary 02 is the palliative setting. And this is going to be a large trial aiming to recruit more than 1,000 patients with biliary tract cancers, cholangiocarcinoma, and gallbladder cancer, and similar or same as in the phase 2 setting, combining gemcitabine, cisplatin, and rilvegostomig, and comparing it to the standard of care with durvalumab, and also similar to what we are already used to. After 8 cycles, this will be switched to a maintenance setting with either rilvegostomig or durvalumab. Now, in the palliative setting, but moving towards the targeted therapy, surprise, we are encountering rilvegostomig. We are here in the HER2 positive patients, and you may know that the antibody drug conjugate trastuzumab deruxtecan has been approved by FDA, but not by EMA for solid cancers, including biliary tract cancer. And ADC is moving into the first line of therapy, and it's being combined in the Destiny-BTC-01 trial with rilvegostomig. It's a 3-armed trial, the first being the actual combination arm TDXD plus rilvegostomig. Then there is an arm B, also an experimental arm, as TDXD monotherapy, which I found a bit surprising, to be honest. And then there's arm C, which is the standard of care arm, gemcitabine cisplatin in combination with durvalumab. Similar, the Horizon BTC-302 trial will be a trial also addressed at HER2 positive patients. We were happy to see that zanidatamab was approved not only by FDA, but also by EMA last year for patients with progressed HER2 3 plus biliary tract cancers. So we have this as a second and further line options already. But of course, it's in the intention now to move this to the first line of therapy, and Horizon does exactly that. It's combining zanidatamab plus standard of care, comparing it to standard of care, including both IHC3 plus and IHC2 plus ish positive patients. The primary endpoint, to my knowledge, however, will only be in the IHC3 plus population. The primary endpoint, despite being a phase 3 design, is going to be the progression-free survival, most likely owing to the fact that we have the approval of zanidatamab already in the second line. There'll be a lot of crossover in that sense. What is also important is that if you want to include patients into this trial and you do not have the results when they actually start the systemic therapy, this is not a big issue for the trial. Patients can have received a maximum of 2 cycles of standard of care chemotherapy prior to the inclusion. Now moving to FGFR2 fusions, which, as you know, are one of the largest groups of genomically largest groups of intrahepatic cholangiocarcinoma patients. We have 2 approvals in Europe here for pemigatinib as well as for futibatinib. And you may remember that both of these drugs were intended to also be tried in the first line of therapy. However, those phase 3 trials closed prematurely because there were a lot of enrollment issues. There's one trial that is currently recruiting for futibatinib. It's a phase 2 trial, but it's basically using different dosages of futibatinib. 20 milligram is the current standard. And in the Phoenix CCA4, there's also one arm that's reducing this dose to 16 milligrams. There's been some additional data that's been reported by the end of last year for an additional FGFR targeted therapy. This was a refocused trial, and lirafugratinib is the drug name. Some of you may know it from the National Relife 408. And finally, we've seen some more data from this trial. It actually took a while for the company to report. And you can see here that in the cross-trial comparison, it compares very similar to what we've also seen from the futibatinib trial, and these data being in FGFR inhibitor naive patients. And the reason I'm showing you this is that this trial also included a cohort of a bit more than 50 patients that had actually already received a prior FGFR inhibitor. And this is exactly what is the patient population that is addressed in the first 308 trial. And first 308 is not using lirafugratinib, but it's using tiningotinib, which is a pretty broad multi-tyrosine kinase inhibitor with a strong FGFR activity. And the patients who were recruited into this phase 3 trial must have received chemotherapy. They must have received, and this is what makes this trial really special, they must have received either pemigatinib or futibatinib, not both, and then they can be recruited upon progression into this trial, and they will receive either tiningotinib or physician's choice of chemotherapy. It's a 2-to-1 randomization, and this is actually recruited quite well, and the recruitment is estimated by the end of this month. So if you still have patients that you want to include into this trial, you probably have not too many weeks left to do so. Moving to another large genomic subgroup of our intrahepatic biliary tract cancer patients, the one that harbor IDH1 mutations. Many of you will be familiar with the Clarity study that has basically implemented ivosidenib in patients with IDH1 mutations that progressed on previous chemotherapy. This was based on phase 3 data on the primary endpoint of progression-free survival, and we already know that we can expect from ivosidenib a disease stabilization, but we hardly ever see actually tumor shrinkage. And these were data that were recently also supported by the analysis from the PROVIDE trial, which was a phase 3b study, also presented by John Bridgewater, who you'll hear later on today. And similar to all the targeted siblings HER2 that we already discussed today, also evozinib is trying to move into the first line of therapy. And this is a trial that is designed as a phase 1b/2 trial that aims to combine in the first line of treatment evozinib with standard of care, gemcitabine and durvalumab. And this trial has just now completed its safety lead-in phase. I think it recruited 6 or 7 patients thus far. It's passed all the safety analysis, and it's now entering the expansion phase. There are some notable inclusion criteria. The patients that are included into this trial must have had their surgery at least 6 months ago, and there must be at least 6 months since completion of the adjuvant therapy. And again, since it's not so easy to include patients into targeted first-line therapies, this trial allows a maximum of 1 cycle of first-line chemotherapy prior to inclusion into this trial, combining evozinib and a standard of care. And of course, as always with evozinib, we have to write an ECG because the QTc should not be prolonged. So moving towards new targets, I think one of the interesting targets from recent 2 years has been Claudin-18. If you're also treating gastric cancer, you know that we have approvals for Claudin-18 targeted antibody, which is zolbetuximab. And there are also some initial analysis reporting Claudin-18 expression also in biliary tract cancer. And the current estimate is that around 5 to 6% of all biliary tract cancer patients actually have Claudin-18 expression according to the cutoff and the assessment that is being used in gastric cancer. So is it going to work as a target in biliary tract cancer? I don't know. For zolbetuximab, I have to admit there have been a bit of disappointing results in pancreatic cancer, and in many ways, sometimes pancreatic cancer is a bit similar to biliary tract cancer, but maybe this is too pessimistic. But the combination of zolbetuximab plus gemcitabine and Abraxane, which is the standard first-line therapy for pancreatic cancer, versus gemcitabine and Abraxane, did not report positive results. Nevertheless, there are different activities ongoing in the field of Claudin-18 to positive biliary tract cancer, especially also focusing on antibody-drug conjugates, as well as bispecifics, so going a little bit beyond the zolbetuximab course of action, and maybe this will be opening up some new avenues. But again, I think it's going to take a while until we see actual results. Here, these trials are currently recruiting. Last but not least, another antibody-drug conjugate that might be of interest is, I always have problems pronouncing these names, datopotamab deruxtecan or Dato-DXd. It's a TROP2 targeted antibodies. There have been some very good results and also actually approvals, for instance, in breast cancer for TROP2 targeting antibodies. So let's see how it's going to perform in biliary tract cancer. I'm mentioning this target because there is a sub-study aiming to recruit 40 patients that is specifically dedicated to biliary tract cancer patients who will receive Dato-DXd as a monotherapy, and it's currently recruiting. So this is going to be my last slide before I will actually hand over to John. And John, you will have to tell me when I have to move to the next slide for you. But I asked a colleague from who's leading one of the big phase 1 units actually in Madrid to share the portfolio that they currently have for biliary tract cancer in second and further line at this phase 1 unit. And I just wanted to share it with you because I found it quite interesting, and I think it kind of might give us a glimpse to where this field is moving, at least in Europe. You can see here that there are a number of trials in this unit ongoing that are using bispecific antibodies, and another 2 trials using antibody-drug conjugates. So maybe just a glimpse into the future, and we will see what we will hear from it in the next couple of months and years. And with this, I would like to ask John to actually come on and discuss with us the Sapphire platform. John, are you there?
Yes, can you hear?
Yes, perfect. I'm glad.
Oh, okay, sorry, I had to go and find another computer, which I've done. So good afternoon,
No
everybody.
worries.
Very much, Anna. And so this is the disclosures. Next one. So this is the Sapphire and ABC10 family of studies and other studies that have sprung up consequent on ABC10, and it will become obvious why this has happened. So this is Sapphire ABC10. You will all know this. So this is our big academic first-line advanced disease study. And in fact, the numbers are slightly different. This is the UK version, but it's running in UK, France, and Belgium. And we are screening, and it's essentially, it's in 2 bits. There's a screening bit and a treatment bit. And the screening bit is up on the top left here. In fact, this is n equals 400, supposed to be the UK. We will probably get to 400, but France is doing somewhat better than us. And France has screened probably again about almost 50% more patients than we have. The total number has increased from 800 to 1060. So fortunately, we're able to continue screening up to 1060 patients. So that in itself, I think, is an achievement for biliary tract cancer. The first bit of it is the profiling, and here in the UK, we send all n equals 400 all to one lab. Our testing is centralized in the UK, tissue testing is centralized, and it's all going to one lab, which happens to be at the Marsden, and they will do standard RNA and DNA profiling. And then also we have simultaneous Guardant for all patients. In France, this is done at one of the 2 commercial providers. It's Sequoia in the north and Origin in the south. And that's not a targeted panel. This Marsden thing is a targeted panel. In France, they have whole exome. And so we will have a huge amount of genomic data on 1,060 patients with advanced biliary tract cancer. That in itself and the comparison of these 2, which is better, I'll give you a bit of a hint of that in just a moment. Then if you do have an alteration, and I'll show you how many do in a second, you get one of these treatment arms. You're randomized to receive targeted therapy or continue standard of care and then crossover. And to date, the study's going well. We will still, despite increasing screening to 1060 from 800, we will still probably finish about 1 year ahead of time. Next slide. So this is how it's gone. So far, we're up to 629. This was, in fact, I can't remember how recent this was, but it's going very well. 483 patients have been reviewed in the MTB, and almost 90% of people have some kind of profile. So 98% of people we have some kind of profile on. We can say whether they have something we can target or not. You will see that actually we've found things that we can't target, but maybe other people can. And we've identified a target in 29%, so slightly less than we thought. And we've randomized 72 with a number still pending. We've lost a few more patients than we thought we would at this stage. So we've lost about a third of patients, mostly through disease progression. And then there are a whole load of other reasons why people. So that's slightly disappointing. We thought that would be a bit better, but all in all, it's actually going reasonably well. It's going reasonably well. Next slide, please. So these are the targets we found. So it's pretty much as expected, and about, as I say, about 30%. And so these data will be incredibly useful for planning future ways of testing patients and what to expect and what to tell patients. Next slide. So in addition, so this is the standard core protocol I've shown you on the left here, which is SCAT2 type of target, and then you're randomized between standard of care and the maintenance molecular targeted therapy. But in addition to this, several sub-protocols have sprung up because we found other things and didn't know what to do with them. So one of these being IDH2. So IDH2 I've always regarded as the poor sister of IDH1, and you know, there is a drug for it, and the drug should work. The biology is identical. It's actually not identical, but it's very, very similar to IDH1. So it should work in a similar way to IDH1. And so, Servier have finally agreed to support a vorasidenib study. So a phase 2 vorasidenib study of roughly 30 patients is currently with the IKF in Leipzig. Is it, Anna? I think it's
Frankfurt. Close.
Frankfurt. It's currently with the IKF, and hopefully we'll get that started in the next 6 months or so. We've already identified, I mean, this all happened because we've already identified something like 15 to 20 patients as part of ABC10 screening. The BAT1 study, yes, there was that was really promising. There was a BAT1 drug which could have used, but actually Californian biotech took a big hit when America decided to be American first, and that's all gone quiet. Californian biotech is still with us though in the RAS study. So RAS, about 20% of all our patients. I think I've got a slide on that a bit later. The Hyperion is a radiation study. That's somewhat with the MD Anderson, that's a bit slow. And we have the second-line severe study, which is using ivonescimab. I think there are further slides about that, Anna, please. So this is the RAS story that we have here. So RAS, somewhere between 25% in the intrahepatics, much higher in the extrahepatics and the gallbladders here. But really, you know, potentially the biggest target that we have. Next slide. And next slide, please, Anna. Here we go. Yeah. Oops. Yes. Well, this will do. We can stay here. So in the next bit of information will tell you that actually one of the most useful things is to have had Guardant in Saphir ABC10, and in the first 200 patients, the hit rate of RAS is about 21%. So that's pretty much what we expect. That's very reassuring, and so we reckon we can recruit very well to any RAS study as an offshoot of Saphir ABC10. I mean, the big plus really is that, you know, these patients have all been screened. There is already clinical trial level data on these patients available. The amount of work you have to do is pretty small. So it's quite attractive for anybody to move in here. Next slide. Yeah, and Kvande and Revolution Medicine have promised us to this drug, which I think has got a name now, beginning with D, which I can't remember, but it does, and so hopefully we'll be able to use what should be an incredibly effective drug. These are the lung and the PDAC data. Next slide. So this is this is how the design is going to happen. So patients who progress or come off the core protocol here will receive a single-arm phase 2 study using the Revolution Medicine drug. Patients from outside of Saphir ABC10 can also be admitted, although I think the majority of our patients will come from Saphir ABC10. We will not have difficulty filling this cohort. Next slide. So one of the other studies that have come up as a result of this is, in fact, an adjuvant version of Saphir ABC10. We've talked about this for a long time, and we've always scratched our heads when it comes to whether the numbers will work. But Julien Edeline is leading both this and the RAS study, and the intention here. And so these are all the historical studies, and the intention is to use some kind of targeted therapy as adjuvant. Next slide. Somehow intensify the very poor results that we have using targeted therapy. Targeted therapy as adjuvant in other cancers does work well. Okay, and here we have breast melanoma and non-small cell where they've used targeted therapies, and actually, you know, it's probably better than chemotherapy, which is very reassuring. So it should be a positive thing to do. Next slide. So this is what's coming. This is Saphir Impact, already funded in France, already funded through Europe by a tract. And I'm literally in the process of just doing the response to Cancer Research UK funding in the UK. So hopefully we will have three. This is going to be genuinely a pan-European study. We've only managed to get 3 cohorts for this: the FGFR, the IDH1, and the HER2, supported by Tahe Sevier and Jazz, respectively. This hopefully will start at some stage in the next 6 months or so. So this is really, you know, going to be a very, adjuvant studies in biliary tract are always challenging, but I think as Actica demonstrated, we can do them. We can do them across Europe, and the approach of doing it across Europe is one that has worked very well for Actica. So I think this will work. And targeted agents in biliary tract, I think you know, deserve testing properly. Next slide. So I spoke about this earlier. So this is again, Julien has really done well here. So this is his second line study. So this is post progression on Saphir ABC10. Originally, this arose because all the people who didn't have an actionable alteration in Saphir ABC10 complained bitterly, said, "What are you going to do for me? You know, I'm the unlucky one. What are you going to do?" So we developed this. So this is the second line study using evonestinib versus FOLFOX, 2-to-1 randomization. Julien's recruited 3 or 4 patients already. We will open in the UK in about 6 weeks, and hopefully that will come through. And then I think my last slide. Yeah. So this is the IDH2 study that I spoke of earlier. So this has got a number of names because essentially everybody had the same idea. So this is Christoph Rodeberg is going to lead it. I think I can't remember which one of these names is his, but we called it ABC14. But it's the same thing. Ostensibly 23, although I imagine we will probably, based on the number of patients we're actually finding as part of Saphir ABC10, it'll be more than that. It's already CR UK approved, and we will get that started. This is a very, very simple single-arm phase 2. We'll get that started very soon. So those, I think that's my last slide, isn't it, Anna? Yeah, brilliant. Thank you very much.
Looks like the last slide. Okay, perfect. Thank you so much. Very much looking forward to discussion now. But before we dive into the discussion, and I can finally unshare my screen here, For those of you, and I hope there are not many of you, have not yet joined the Precision-BTC network, this is how you can do it. You can also just write us an email and we can send you the instructions. You have to join the network, and then please also join at least one working group. If you join a working group, also join working group 3, because that is the one that's most centered on the precision medicine, also in biliary tract cancer patients. And I would also like to invite you to join us in beautiful roads, actually. Quite soon in June, we will have abstract deadline that maybe, Rocio, you can say it again when it's going to be, but I think we are opening abstract submission. It would be great to see as many of you in our Precision-BTC conference on roads. And with this, I would like to thank you for your interest. Looking forward to our discussion.
Helen has a question.
It's for Professor Bridgewater. John, what is your opinion on using targeted therapies in first line for patients who are molecularly profiled and obviously have a targetable mutation?
I mean, I think that's where it's going to go. In fact, we tried to do this before and it failed because we couldn't get the testing done in time. At some time in the future, we will have testing that is instantly available, and we'll be able to do this. And it makes sense, you know, to the later you leave a tumor, the more biologically disordered it's going to be. So to bring it forward is hugely sensible, both in terms of adjuvant and neoadjuvant, for instance. But at the moment, it's a question of the testing.
Yes, and we know how difficult that is for all of us at the moment. So thank you for that.
Okay. Paul, your hand is up.
Thank you. I had a question about some of the gene faults which are less commonly seen and may be part of perhaps basket studies which are pan-tumor. So we've seen recently in the UK, we've had NICE assessing larotrectinib for NTRK fusions, and the data that they're using for that is from basket studies. And we're a little bit concerned that it's not very compelling because you've got some groups which respond much better than others. Is this something that we're going to see more of in cholangiocarcinoma? And is that potentially a problem where if we've got some relatively small numbers within studies that they're not then going to be licensed?
I mean, so my impression is that we are learning a bit also from the FDA. I think the FDA are very much open to approvals based on basket trials. I think it does make sense from a patient perspective, because in a rare cancer, it takes us ages, or it's even almost impossible to recruit only in the sole cancer entity. I fully share, however, Paul, your concerns that we cannot simply transfer from one tumor entity to another. We may be in for some good but also bad surprises there, right? So I think basket trials are important. In many instances, in rare genomic subgroups, they will be the only trials that are really feasible in order to also include cholangiocarcinoma patients, at least have the option of getting approvals and drugs into the clinics. What I very much like is, and as John also really nicely presented, are these platform trials, right? Where we are more flexible in adding arms, where we are making the most use of the data that we can. Because if you're only screening for this one alteration, there's so much information that's getting lost. And I think this is really the way to go and a good way forward.
There is a question in the chat. Elene asked if is there any specific procedure or medication that you are specifically looking forward to getting the results from the trials?
Personally, yes, many. If I had to choose one, of course, I mean, we've talked about moving the targeted therapies into the first line in combination with standard of care. So we will have data there, at least for HER2. I think that's something that we can realistically look forward to. Of course, for me, it would be great to have another targeted option for a larger genomic subgroup. And I'm thinking especially KRAS mutant biliary tract cancers. And I do not really think that monotherapy will be the way to go. But I do have hopes for combination approaches. And I wish that at least in Germany, we could have more trials open also for biliary tract cancer. Currently, it's a struggle to get a spot for a RAS mutant biliary tract cancer patient in my country. Maybe John, Angela, do you have any top picks you're really looking forward to?
What I have is a little bit of a concern of something that I am now seeing with both HER2 clinical trials in the first line setting happening kind of at the same time, with both really struggling to recruit. So I wanted to know, in my practice, what I am doing is HER2 locally for all the patients, but still I am not really finding patients that are suitable. Is that the same case you are doing, or are you following a different strategy or something we could maybe consider? Because I will not want the FGFR story to happen again.
Yeah, I think it's an issue, right? I think HER2 testing is very much in the eye of the observer, or more specifically in the eye of the pathologist, right? It's not an easy assessment, and it's not so easy to standardize. And I think Jazz Pharma for the Horizon trial, they are really trying to urge investigators to send in the tumor materials for central testing. And I think for a difficult histopathological immunohistochemistry-based marker, central testing makes a lot of sense. And I'm always puzzled too, Angela, because I see that our colleagues in France, they are recruiting into HER2 trials many more patients. Also on Saphir, I think many more patients than I see to turn out positive in my practice. So I genuinely think there is a differences in assessment by pathologists, and we need centralized testing for the more difficult inclusion markers. John, what's your take on that?
So it's a question of education amongst our lovely pathology colleagues, really. It can be done. They've done it for breast, they've done it for esophagogastric, to bring in routine HER2 testing. In the UK, I think the stick to do this will be, touch wood, the approval of zanidatamab by NICE, which hopefully will occur in the next few weeks or so. And when that happens, then HER2 testing should roll out completely as reflex, and it requires that kind of stick. It's because everybody in the UK is obliged to follow NICE guidance. It's like the closest thing we have to the Bible, and so I think that's it. Only when that happens will it become easy. Until then, it's a question of me shouting at my histopathology colleagues. I love them all, but sometimes they don't do what you want.
Hello, Arman.
Hello. I would like to ask a question about ivonescimab after Saphir. So I think it will be a IORI challenge. So will it be possible to combine with FOLFOX, because it's used with combination with chemotherapy in lung cancer, and we already use with FOLFOX immunotherapy in gastric cancer and esophageal cancer?
Yeah. So we had this question is an excellent question, and it's come up with every single trial application for funding that this idea has gone through. and the answer was that we felt that Angela, forgive me, low bar. Okay, FOLFOX is a very low bar, and consequently, it would be reasonable to use it as by itself. Put it the other way, is how much would you gain by adding FOLFOX in combination? Something pretty modest, we reckon. So it's that sort of conversation. I think following a different discussion, we could have easily gone in for combination treatment. I don't think there's a lot in it.
Thank you.
Perfect. Any more comments? Any important trials you want to advertise that we forgot to mention, we should have mentioned? If not so, I would say we can close to today's webinar. And Angela, I think the next one is probably going to be in September again, right?
Yeah, probably, yes. Yeah, we will arrange everything every 6 months.
Okay, great. Well, thank you all for joining. Thank you, Rocio and Sam, for organizing. And we see you on roads.
Thank you. Bye-bye.
Thank you for
Bye.
organizing and preparing this.
Bye-bye.
Bye.